Hello again, Year 9. This is Mrs. Cowlin with Lesson 4 about transport across membranes. This lesson is an introduction to osmosis, including some information about osmosis in animals. So you have 3 objectives from this lesson. The first one is to know that osmosis relates only to the movement of water molecules. Secondly, you will need to know the conditions required for osmosis. And thirdly, you will need to know some effects of osmosis on animal cells. As this is a YouTube video, you are free to pause, to make notes, and to rewind if you need to check something. There are some key vocabulary terms and some phrases in this video which are in red, so that you know that that is what you need to focus on and you will need to copy down. So let's go. So first of all, osmosis is similar to diffusion because it involves the movement of particles again. However, osmosis is only involving the movement of water molecules, where diffusion is the movement of gases or of solids dissolved usually in water. This is actually talking about water movement, and it involves the movement of water from an area where water molecules have a high concentration to an area where water molecules have a lower concentration, and once again it involves movement across a partially permeable membrane. If you remember, a membrane can surround either an organelle such as a nucleus or a whole cell, and permeable means that it is able to have substances moving in or out of it. When it is partially permeable, it will only allow some substances to move in or out, not all of them. So here we've got a beaker, and the beaker is split down the middle with a partially permeable membrane. So you've got 2 different solutions on the left and on the right. On the left hand side you have got a solution where there's fewer molecules of sugar and more molecules of water. And if you have a look, the sugar molecules are orange and the water molecules are red and white. So you can see that there's more of the red and white water molecules and less of the orange sugar molecules on the left. If you look on the right hand side, the situation is reversed. So you have far more sugar molecules, far fewer water molecules. So if we're talking about concentrations, we're talking about it with regards to water here, and we'll say that there is a lower concentration of water on the right hand side than there is on the left. And the concentration is how many molecules out of all the molecules on that side. Now I'd like you to pause the video here and make a prediction as to which direction the water molecules will move. Okay, so here is the same container after some time has passed, and the water diffuses from the left to right to equalize concentrations, and the diffusion of water is called osmosis. Okay, so it is a type of diffusion, but it is only involving water, and obviously the situation will tend to equalize things. And in this case we're looking at the red and white water molecules, and they will be moving from the left to the right to equal out the concentrations. The sugar molecules will remain in the same sort of place. Okay, we're not worrying about the sugar molecules, that's regular diffusion. We're just talking about osmosis, which is the diffusion of water. Now, why do we need to have osmosis? The cytoplasm in a cell, which is the clear jelly-like substance, you sometimes will think it's sort of empty space when you're looking on a slide, but it's actually a clear jelly. It's mainly water, but it also has dissolved chemicals. Chemicals are mainly sugars that come from digestion in animals or photosynthesis in plants, and salts. And the solution that results from it is the place where most of the chemical reactions will occur. The mixture of sugars, salts and water is kept inside the cell by the partially permeable cell membrane, and when we're looking at diffusion, we're thinking about the concentration of the salts and sugars. When we swap to the word osmosis, we're looking at the concentration of water. And obviously for the reactions to continue to carry on efficiently, the cells need to maintain a fairly standard mixture within them. Now, there are 3 pieces of vocabulary that you need to learn for osmosis. They're very similar. They're isotonic, hypertonic and hypotonic. The last 2 you need to look at very carefully for the spelling. And you might have heard of an isotonic sports drink. I think Powerade is one. There are others available. And these are sports drinks that athletes have when they have been playing in a football match or running a race, because through the process of being active, they will have sweated, they'll have lost water, they'll have lost sugar and salt, and they need to rebalance their body. And the solution in the bottle is isotonic because it matches what the body needs. So it is at the same concentration, and isotonic means the concentration of solutions on both sides of the membrane are equal. So iso is equal. Hypertonic is when the concentrations outside the cell are higher than inside the cell, and hypotonic is when the concentrations outside are lower than inside. So the opposite, hypertonic, hypotonic are opposites. And osmosis is only the movement of water molecules. And we're talking about the concentrations, we're talking about net movements. We're not looking at the movement of one individual molecule, because that can move in any direction. We're looking at the overall change, the overall balance. So let's look at this in action in a real life context. And we're going to think about blood. Now, blood has several parts. It has the plasma, that is the fluid containing the white blood cells, the red blood cells, the platelets, and also the dissolved substances. And your red blood cells are cells, obviously, so therefore they are able to take part in the process of osmosis. Normally, a red blood cell is biconcave. That means both the sides curve in towards the centre, and that gives it an increased surface area, and that is what they look like when they are in balance. Now, if you were to put those cells into different solutions, you will get different results. So if the cell were to lose water, for example by being put in a concentrated salt solution where there's lots of salt, not so much water, water would move from the red blood cell into the solution outside it to try and even things up, and at that point the cells could shrink and they would be described as dehydrated. And you've probably heard people say that they need to drink some water if they are dehydrated, and that is why, because it literally damages your cells. They will lose water, they will shrink, they won't function as effectively. And the opposite situation, if you have too much water around the outside, then the cells will take the water in, and initially they will just swell, they'll get a bit fatter, but obviously there is a limited capacity because the membrane is a certain fixed size. It cannot just grow and accommodate this extra volume, and eventually it could actually burst, which would be the end of the red blood cell, which would affect the carrying of oxygen around the body. So this is obviously not a desirable outcome, and for that reason your red blood cells, rather than just being on their own as the main part of the blood, are actually within plasma, and the plasma actively regulates the environment for the red blood cells so that they neither shrink nor expand. Now we will cover plant cells in a separate lesson, because there is a lot to learn about plant cells, and there is also a required practical that involves plant cells. So for today, that is the end of what you're learning about osmosis. You have got a worksheet to complete that is on Padlet. You can submit it via Teams so that it can be checked by your teacher. There is also a quiz to complete on Teams. Thank you very much, Year 9.